we're DNA Awesome, and we're here today to tell you a little bit about receptor tyrosine kinases and their dimerization process. Receptor tyrosine kinases are enzyme-linked receptors. That means they are transmembrane proteins that receive signals from the environment to cause a subsequent cascade of cell actions while also functioning as enzymes. The binding of a signal molecule activates the receptor's enzymatic activity. In terms of structure, receptor tyrosine kinases have an extracellular portion that binds to ligands. This is called the ligand binding domain. On the intracellular side, the receptor has an enzymatic domain that acts as an enzyme when the ligand binds to the extracellular side. Receptor tyrosine kinases are extremely important because they help regulate cell growth, differentiation, and survival. Receptor tyrosine kinases are unique because they contain tyrosine on the intracellular enzymatic domain. When activated, the enzymatic function of these receptors is the transfer of a phosphate group from ATP to intracellular proteins. Phosphorylation of various proteins can carry out messages in cell through signal transduction. The receptor tyrosine kinases come in pairs and act as dimers. When a signaling molecule binds to a receptor tyrosine kinase on the extracellular side, this causes neighboring RTKS to associate with one another and form what is called a cross-linked dimer. Receptor tyrosine kinases must act in pairs because that is how enzymatic activity is initiated. Once active, each tyrosine on each RTK picks up a phosphate group from ATP. Different proteins will then combine and attach or dock themselves to the tyrosine residues to either activate or inactivate the proteins. This can act as the initiation of a long and complex signaling cascade that can sometimes reach the nucleus to upregulate or downregulate the gene transcription, thus affecting the production of proteins. Receptor tyrosine kinases can bind hormones such as insulin, which is a pivotal process in metabolism. The insulin receptor plays a key role in regulation of blood glucose homeostasis. When the glucose homeostasis is interrupted in the body, this may result in clinical diseases such as diabetes and cancer. Because RTKs often regulate cell growth, when they do not function properly, cancer may result. Many chemotherapy drugs target RTKs. One example is Herceptin, which is an antibody used to inhibit a specific overexpressed RTK in many breast cancers. In conclusion, receptor tyrosine kinases bind extracellular signaling molecules to turn on enzymatic activity inside the cell. This can then result in a series of enzymatic reactions and signal transduction within the cell to potentially alter gene expression at the level of transcription.